I request all the people to kindly mute their phones. Good afternoon, all. It is our pleasure to welcome you all to the 14th R.N. Chakravarti Memorial Oration. We at the Department of Experimental Medicine and Biotechnology are conducting this oration every year to commemorate our esteemed late Professor R.N. Chakravarti, whose vision was to inculcate scientific aptitude in future generations. Now I invite Professor Dibyajyoti Banerjee, Head of the Department of Experimental Medicine and Biotechnology, to officially welcome you all.
So, good afternoon, everybody. It's my proud privilege to welcome you all in this 14th R.N. Chakravarti Memorial Oration. I formally welcome Professor Sanjay Jain, today head of the institute, Professor Madhukullar, Veena ma'am, Divya ma'am, Professor Sushmita Ghoshal, and all esteemed faculty of this institute. A warm welcome to Professor Raghavan because he has very kindly accepted this invitation from his very busy schedule, which will be understandable when we will introduce him. So, dear friends, this is I welcome you all. Not just for sitting, it's a really opportunity because here we will know something from an original worker. So, a warm welcome to you all. Also to Prasenjit, I think it is the Devotjit. Thank you. Sapna.
Thank you, sir. I request Ms. Parul to kindly present a bouquet to our orator, Professor Satish C. Raghavan, Professor, Department of Biochemistry, Indian Institute of Science, Bengaluru, as a token of respect. I request Ms. Neha to kindly present a bouquet to Professor Sanjay Jain, Dean Research PGI, who is our chief guest. May I, may I now invite all the dignitaries to light the lamp and start the function with the blessing of Goddess Saraswati Ma.
Sa sa re sa ni ni sa ni pa pa ni ma 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 ga ma ma ni dha dha sa sa pa pa ma ni ma 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 ga ma ma ni sa. माँ सरस्वती शारदे माँ सरस्वती शारदे इस अज्ञान उदय से इस अज्ञान उदय से पीड़ा तार दे तार दे तार दे माँ सरस्वती शारदे माँ सरस्वती शारदे.
Thank you. May I now request Professor Sanjay Jain, Dean Research, to kindly say a few words in Professor R.N. Chakravarti's honor and about this oration.
Distinguished speaker of the day, Professor Raghavan, senior faculty colleagues, PhD scholars, students, and my dear friends. In fact, it's a matter of great honor and pleasure for me to welcome you all on this great oration in the name of Professor R.N. Chakravarti. I think who is ingrained in our DNA of PGI, and possibly that's the reason that we have chosen the speaker as who works on DNA repair. So let me put it very straight that the orations are not ceremonial, but they are acts of remembrance. Actually, all the orations give us time to pause, to think, and to possibly pay our homage to the shoulders on which we stand today. Please remember that Professor R.N. Chakravarti, he was born in 1925, and I think he did his MBBS in 1948. The time when he started his career in the field of, I would say, research, in medical research, those were the times of 1960s and 70s. That was the time, please go back to the history, that India was developing. The health infrastructure was very meager. We were developing our health system to deliver it primarily. And I would say at that time, research in the field of health was absolutely at its infancy. At that time, Dr. R.N. Chakravarti was possibly one of those rare physician scientists. Maybe at that time, actually nobody would have even heard the term, but he was one of the best physician scientists India could ever have. Again, I would like to stress that no institute is either built on the so-called bricks or the wealth. It's always built on mentorship, guidance, and the original research. And Professor R.N. Chakri was one of them. I never met him, I've never seen him, but the stories which I heard from his students, Dr. N.K. Ganguly, Dr. Majumdar, and we have two, Dr. Khullar and Dr. Veena Dhawan. Is that right? Yes. The stories what we heard are really, you have to really imagine the type of person he was. He built something which people can even dream of. They were dreaming and he was doing, he was performing, he was executing. He built the Department of Experimental Medicine not as a second to the clinical departments. He built it as a discipline. And there were very few in the country at that time. He was the only one who started the animal research at PGI, developed many models, including rhesus monkey model on atherosclerosis and hypertension. And we are we are getting the fruits out of it. We talk of translational medicine, molecular medicine, you name one advanced technology in medical research, but the seeds were sown decades ago by people like Dr. R.N. Chakravarti. So, in fact, it's my humble tribute to him particularly, and nothing great could have happened to PGI than to invite a person who exactly follows his footsteps, and that's Dr. Raghavan. So, it's my great, I would say, pride. 30 or 40 minutes are going to be stimulating, and we'll have one-to-one interaction. Thank you very much.
Thank you, sir, for such a wonderful introduction to a great legend, Professor R.N. Chakravarti. May I request our head of the department, Professor Dibyajyoti Banerjee, to kindly introduce Professor Satish Raghavan, our orator of the day.
So, it's needless to emphasize that I'm very happy to introduce Professor Raghavan, who has become a friend, only a day's friend, of course, but I think it will continue. Now, although he's a great scientist, but like all great scientists, he started his career very humbly from Payyanur College, then Calicut University, Entomology Division, then Benares Hindu University. I think that time Dr. Maharani was there when you were there. I was also coming in that time, but not exactly well. So then, Los Angeles, University of South California. Then coming back, many people go, come back, and a thing what they do, he has also done. You have to focus this. after coming back, a Cell paper, 2012. This citations around 4419. Now, newspaper is saying that USA companies are trying to buy it, and it is relevant in 2016. I think 2026. This is the 15 paper, right? And it is relevant in 2026 by independent editorial who is quoting. Perhaps Dr. Satish has not noted this. This is 26 reference to the Indian science report. Now, this means, and another thing, that we all buy chemicals from companies, but we never meet the persons who have invented it. So, small molecule inhibitor of DNA repair, one of them will be very, very lecture. So, I think this is the introduction. What I mean that we must visualize an original invention. It is customary to say this long list of awards I am saying, but the hard work what I am showing you, that this is the thing, that Dr. Raghavan is the recipient of several awards, including Shanti Swarup Bhatnagar Prize 2013, rightly, the Leukemia Research Foundation USA 2010, the Nazi Reliance Industries Platinum Jubilee Award 2015, Indian Society for Cell Biology ISCB, SP Rai Chaudhary Lecture Award 2023, Society for Biological Chemistry India, Ramarao Lecture Award 2024, J.C. Bose National Fellowship 2025, Fellow of all the science academies normally, but the most important thing is that we will be here an original researcher whose products we use. Thank you very much. I welcome Dr. Satish. Wonderful. Yeah.
Okay. Good afternoon, everyone. Also, thank you, Professor Banerjee, for the kind introduction. And it's very nice meeting you, Professor Jain. Um, Let me also thank the committee involved in choosing me for this prestigious oration lecture, Professor R.N. Chakravarti Memorial Lecture. I mean, in fact, I heard a lot about him from today morning onwards, and I was amazed to hear all those stories. From that early on, somebody had a vision to have medical research and the basic research coming together and establishing a department like this, and then nourishing that, particularly a time when probably it was not very conducive to have a clinician working together with the basic researchers. So that's something amazing. Also, it's great to know that he initiated the animal studies in this institute, and naturally, the current generation are getting benefited out of that. Just to say, I happen to be also the coordinator for Central Animal Facility in IC Bangalore last 5 years. It's just a coincidence. I thought I'll just mention. So we are also, I mean, we have an animal facility there uh, from 1950s, but that was like something very little. In fact, it was not a facility. People used to do animal research next to their lab and all those things. See, the kind of ethics we are now, of course, it's very important and worried, and then we are following. Those things were very limited at that time, right? So I happen to coordinate the animal facilities there. So again, thank you very much, and it's a honor for me to deliver this lecture. Um, Once again, let me take this opportunity to thank Professor Banerjee, Sheetal. She happened to be my third PhD student a long time back, and all the faculty members of the department for inviting me to this place and choosing me for delivering this lecture. And happy to see all of you here, and hope I'll not bore. I have a lot of slides, But I will ensure that, I mean, I have to, actually, I have to skip some of the slides. I kept a little too many slides. So in between, I may be skipping, but then I'll try to cover some extent my journey after I joined IISc as an independent researcher and what we have done. And then towards the end, I'll talk about the discovery of CR7, which happened long time back when Shidal was also part of the lab. But after that, we have now a new generation inhibitors, which we are hoping that we'll be able to take it forward. CR7 had many limitations. I'll talk about that. But that was actually the original finding. From there now, we have moved on quite far. So with that, the title of my presentation is something like DNA double-strand break repair, mechanistic understanding to relevance in cancer therapy. So now, I don't know for this audience, DNA, of course, genetic material and DNA damage, I'm sure everybody would have heard. And then, oh, okay. Is it the correct? Okay, I have to use it. Yes. So this slide kind of summarize in case if you have forgotten about the DNA and DNA damage and DNA repair. This is somewhat almost everything what is known till today. Some sort of a summary. I think I'll close this because I'm seeing here my own picture. So it's better. Yes. So now, as you know, genetic material, which is of course present in our chromosome, whether it's normal cell or cancer cell, right, can get exposed to varying different type of DNA damaging agents, which could be, as shown, something like that, X-rays or ionizing radiation like gamma rays, X-rays, or UV light. These are all called external agents. Then, chemicals, pesticide, we always talk about. But actually, the fact is, most of the DNA damage in our body, in our cell, is induced by internal factors itself, right? Replication induced error and free radicals. I mean, I'm sure you would have heard about it. So all these type of damages, which could be at the level of base, if you remember, I'm sure there will be some students as well. So hopefully you'll remember the DNA structure where base, nucleotide, right? And also the two nucleotides are connected through phosphodiester bonds to form single-stranded DNA, and then finally double-stranded DNA connected through hydrogen bondings, all those things, right? So the fact is the DNA damage also can be at the level of base, nucleotide, or at DNA strand level. But fortunately, we are all evolved with the DNA repair mechanisms, right? With or without our knowledge, this DNA repair keep operating in our trillions of cells, what is present in our body, right? So therefore, none of us are normally bothered about it unless somebody gets some sort of a problem. You ended up getting into hospital, then people start looking at the mutations, changes, 53, this gene, right? All those things. But fortunately, all of us, the one who claim or we believe we are normal people, healthy people, right? So we happen to have all this efficient DNA repair mechanism in every cell, right? Multiple DNA damage is introduced every day. Okay. So now my talk today is going to be more focused on a particular type of DNA damage called DNA double strand break. Now, so in case if you don't know what is a double strand break, it's very simple. Both the strands of the DNA getting broken in a close vicinity as shown here, right? So nothing, so much to worry about it. So now whenever there is a double strand break like this, then there are three outcomes. Whether it's a normal cell, cancer cell, this is more or less same. Only the level of these activities may change. That's what happened with cancer and other diseases. So normally it gets repaired. That is what we say as DNA double-strand break repair, which means integrity of the genome is maintained, right? The continuity of the genome is maintained. Otherwise, whenever there's a break, if you can imagine, there'll be problems like when replication, transcription, moving on, right? There can be problems. So then that cell can end up in many sort of troubles. So if you look at another outcome is if there's a misrepair, which means wrong ends of the broken DNA get joined. In other words, you know that DNA is organized. DNA is, of course, a genetic material, and in human and other mammals, it's organized into chromosomes 46, all those things we know. right? So sometimes wrong ends of the chromosome get joined. That is what resulting in chromosomal abnormalities like translocations and things like that. Now there are also there is another scenario where there is no repair, no double strand break repair. So what will happen? The broken DNA will get accumulated in the nucleus of the cell. Hmm? So that could activate the cell death pathways such as apoptosis and things like that. Now, if you think about cancer therapy and things like that, which I'm sure all of you will be aware of, whether it's the chemotherapy or sorry, radiotherapy. See, essentially DNA damage in most of the time. Radiotherapy, radiation induces single strand and double strand breaks, right? Of course, other modification as well, right? So then you are bombarding the cell with multiples of 2 grays and then multiple batches and all those things. You essentially kill the cells by inducing DNA damage or DNA breaks. And similarly, chemotherapy therapeutic agents, similar kind of a situation. It induces DNA damage in many cases, right? So that is what essentially today's talk. In case if you are busy and if you want to go today, whatever I'm going to talk will be centered around all of this. In fact, my lab research, about 70, 80, 90% actually centered around these three themes. Hmm? So today's presentation, I will try to talk or highlight some of our findings. And wherever I feel like you are not able to follow, I'll skip those slides and go into the next section. And a lot of work what we do, it is associated, connected with biochemistry, molecular biology, and some extent cancer biology, therapeutics, and all those things. Okay, so as I said, so I'll be talking about these three aspects. Mechanistic insight into chromosomal translocations and other mutation associated with lymphoid cancers, mostly leukemia and lymphoma. And then I'll try to talk a little bit about basic DNA double strand break repair in cancer cell as well as normal cell. And then there are some new findings. In fact, the DNA damage repair research in my lab initiated by Shidal when she was a PhD student. And then finally, I'll try to talk about targeting some of those DNA repair pathways. And there is another molecule called BCL2, which I'm sure all of you would have heard, which is an anti-apoptotic gene, mostly expressed or overexpressed in majority of the cancers. So targeting that for, killing cancer cells. This will be kind of early summary of my presentation. So um, I'm sure you all heard about chromosomal translocations. And, if you look at the early history, I think that's next slide, somewhere around the 60s, right? Janet Rowley discovered or identified first translocation. So that's what the Philadelphia chromosome later it was coined. But that time techniques available, there's no molecular biology, nothing. It was purely cytogenetics by looking under, under microscope, right? That's how it was identified. After that, if you look at it, there were many, many chromosomal translocations identified connected to leukemia and lymphoma. And more recently, if you look at, people believe that even carcinoma and some of the like prostate cancer and few others, there are fusions reported. Arul Chinayan and group actively work on that. Hmm? But then if you look at 80s, so now 60s, 70s, discovery started, it continued. And even now there are new reports, but active reporting of translocation happened somewhere 70s, 80s, and 90s like that. But 80s, 83, 84, 85, that's what molecular biology started, the first experiments in the molecular biology, for example, cloning, sequencing, and all those things happened. So somewhere 83, 84, that's the time people started using DNA sequencing method first time to characterize some of those chromosomal translocation junctions, right? And then see at that time, if you characterize one of those junction, it would be a cell paper or nature paper. Hmm? But now if you do, maximum it will go to maybe blood or something like that, right? Because that time the information generated, everything was like brand new, novel, and things like that. Now, if you look at the historically, after 10 years, that's where, okay, so by that time, there are a lot of sequencing informations were available in the literature. People started looking at all these patient breakpoint regions, this chromosome breaking and joining to other one, and then trying to look at what is the meaning of that. Why would, see, we have such a huge genome, right? 600 billion, 6 billion nucleotides in our cell. right? But if you look at those patient breakpoints region where it undergoes chromosomal translocation, you see that those genes tend to break in a very small region. Sometimes it can be a particular nucleotide, sometimes it can be a region, but it's not just like that randomly anywhere, everywhere. So that's where people like us and many other groups try to understand what is the mechanism, why those chromosomes break in those patients. To start with, of course, it would have been in one cell, because the person whom we refer today as a patient, maybe 5 years, 10 years, 20 years down the back, see, one cell had this change, right? That is what normally we call it as primary hit and all those things. But then, of course, we know, there are secondary, tertiary, many changes, additional mutation, P53, and then epigenetic changes. Everything finally contribute to the tumor development. So now this is one aspect I started work on as a postdoc. First time I got the exposure. We thought of working with 1418 translocation, which is seen in follicular lymphoma. I'm sure all of you may be knowing. And we started asking, why 14 and 18? 18, if you look at a particular gene, BCL2, right? That is where most of the breakpoints are seen. And BCL2 is a huge gene, more than 200 kb size gene. But the breakpoint in the patient is restricted to only about 150 nucleotides. So it's a long story. That's my 6 years of life in this one slide. Essentially, what we found out at that time was this region, major breakpoint region, can fold into something called a non-B DNA structure. Now, you may wonder, what is this non-B DNA? See, I already told the DNA in our cell, it's a double-stranded DNA, which is called BDNA, which is discovered by Watson and Crick, right? 53 and things like that, right? So what we just found out was this region have a lot of single-stranded character. So therefore, we just said that it's not in BDNA. That's the meaning of non-BDNA. We also went on to show that this region can be cleaved or cut by a protein called recombination activating gene, RAG1 and 2. I think some of you would have heard RAGs play a very important role in our immune system development, right? During immune system like rearrangement, the same protein cut there. So this was a new role discovered for this protein, wherein it cut this non-BDNA structure and caused translocation, which was at that time published in Nature. So now when I started my lab, that was one of the first questions we asked, what is this non-BDNA structure? So these are some of the examples. Now I am not going to get into so much of details, because as I said, I have three different sections to cover, but I'll just give highlights and more. Okay. So these are like, depending on the sequence in the genome, or kind of repeat sequence or something like that if you wanted to tell. There could be cruciform triplexes, which is 3-stranded DNA, 4-stranded DNA, which is G quadruplexes, and so on. Hmm. So my first student, Mrudula Nambiar, now she was like senior, Shidal senior. She's a faculty now in ISER Pune. So she showed that the non-BDNA structure, what we talked was something called G G quadruplexes. So now what is the G quadruplexes? When there are structures of guanines in the genome, which can undergo some sort of a pairing by using Hookestein hydrogen bonding and things like that. So think that is an alternate form of DNA structure. Now, later we went on to show that not only, I'll skip that, not only in the case of follicular lymphoma and BCL2 major breakpoint region. See, if you look at the database, this is somewhere done around 2008, 9, 10, this study. So there were, at that time itself, a lot of these translocation breakpoint regions were available. This guy Vijay, downloaded all those things because he was not interested in doing wet lab experiment. But he was good in doing computation. So he found out that majority of this breakpoint region actually possess sequences that support the formation of different sort of non-BDNA structure. Particularly, he was looking at the G quadruplexes. It's about 45-50% of the patient breakpoint region indeed possess those kind of sequence motifs. So that's all. But the journal liked that time. They published it as a cover page article. This is another example. I'm going to skip that. But I talked about Philadelphia chromosome. Janet Rowley discovered 63 or 67, something like that, right? But actually, the fact is, although this was one of the first examples, the mechanism by which BCR and ABL get cut and rejoined during this translocation was unknown. There are certain other studies where one group talked about ALU repeats and all those things. Otherwise, by and large, it was missing. But for us, it was like an interesting case because when we started looking at the region, we found that, see, this arrow mark indicate the patient breakpoint region. There are some small boxes there you may not be able to see, but if you look at those sequence carefully, you can see many of those that region contain many G quadruplex forming sequence motifs. So now with this information in your mind, probably now you can guess what you would have done. And that is what, okay, first, before going to the detailed study, we did something called sodium bisulfate sequencing. Some of you would have heard about this chemical sodium bisulfate, especially if you are doing DNA methylation studies. This is one of the chemical which can which can give specific information about methylation pattern in a stretch of DNA. The only thing what we did differently was we see normally during methylation studies people denature and then treat with the chemical. But here we don't do, because the hypothesis is because of this DNA structure you will have a single stranded region. So we found out that this region can actually possess quite a bit of single strandedness, and that is Elizabeth and Sheetal. I think I have to skip some of these details because these are the actual experiment what we have done, wherein we try to do some gel shift assays, where if you have a control sequence versus the sequence which fold into that structure, it can give a mobility shift on a gel. Like if you show here, see here, so this is I think maybe the other place. See here, this is a C strand and this is a strand which moves much faster in presence of potassium. Potassium facilitates the formation of G quadruplex structures. And then, of course, we did many levels of experiment to demonstrate that indeed it can fold into multiple G quadruplexer structures in the BCR region, which then there is an enzyme called AID, activation induced cytidine deaminase. It's a deaminase which can deaminate cytosine. So now if there are students, if their memory is fresh, if there is a deamination of cytosine, you will have a uracil. Yes, very good. I think many people told. Very happy to hear that. Right? So CTU change in RNA perfectly fine, right? But in DNA, when it happens, it's a mutation, right? So now this enzyme aid, which after deamination, if there's a failed DNA repair, could result into DNA break and translocation and things like that. So that's how we have explained that. And then we went on to work on other translocation. This is something related to diffuse large B-cell lymphoma, where BCL6 transcription factors, Vidya and other people showed that it can fold into many other forms of structures. And this is another example where translocation between chromosome 8 and 14 seen in Burkitt lymphoma where c-MYC breaks. But here what we have found out is something called RNA-DNA hybrid, or R-loop, which is transcription dependent. Again, I'll not get into the details. Later, if you have questions, I'll be able to answer all those things, which can contribute to the fragility and the translocation. This is another case of TOL where BCL11B, another transcription factor, here it get mutated. Okay, and it's T cell specific. And then again, we went on to show that there are multiple DNA forms structures that could contribute to those kind of altered level of expression by mutating it because of the difference in the DNA structures. So now the second part is related to more on the protein called RAGs, which, as I said before, which is normally involved in antibody diversity related aspect, and there it acts as a sequence specific nucleus. Now what is sequence specific nucleus? Which means it's like a restriction endonuclease. I mean, that's the easiest example, right? It's a particularly uh, recognized and coordinate sequence. Same way, during VDJ recombination, so that's that recombination process named, it cuts the recombinational signal sequences. Now, if you understood, while working on translocation, what did we found out? We found out a new function for RAGs, right? It can cleave the DNA structure, altered forms of DNA structure. So that is what That is what was trying to characterize here. Again, I'll not get into too much detail. It's pure biochemistry, where Abni, he was another PhD student in the lab. He showed that even the protein, purified protein, when it cut on structures like this, there is a sequence specificity. If there are cytosins, then both top and bottom strands are cut. Whereas if it is something else, for example, purines, adenine and guanine, this protein cannot touch. And Rupa went on to show that even the neighboring sequence, the flanking sequence, play a role in terms of rag cleavage. There are specific domains required for that. I'll not get into that. get into that. So this is another interesting finding where this is the lab where I worked before, where we found that CPGs, there is a clustering of CPGs at the patient breakpoint region. So we were talking about the DNA structure and motifs, but this is a newer, it's not anymore new, it's published in 2008, but it's a little more recent compared to what was my original study, where we found that CPGs are present at the breakpoint region. And then what was hypothesis is, like what I said, a spontaneous deamination of cytosine could lead to a UG mismatch. And if it's a methylated cytosine, then it will lead to a TG mismatch as shown here, and that could result into translocations and things like that. But now what we did is we tried to look at several of the patient breakpoint region, and we found out that actually not only CPGs, next to that there is something called nonomers. So this is something which I have not mentioned. Normally when rags cut during VDJ recombination, RSS, which means there is a heptamer and nonomer, and then it bind to nonomer, it cut at the heptamer. I know it's a little complicated molecular biology plus immunology, but I think many of you would have studied this, right? So now what we found out is in this translocation breakpoint junction, we looked at more than these 3,000 patient sequences. It's all available in the database. So we found out that 93% of the cases actually have nonomer. And out of that, 74% have both nonomer and CPGs coming together. In other words, the paper which was published previously, we tried to make an amendment, right? So this is all started with in silico work. Then, of course, we did a lot of actual biochemical experiments to show that several of leukemia and lymphoma associated chromosomal translocation, nonomer dependent rag cleavage at CPGs occur, and that can explain those. But now there is a problem. Like if you understood whatever I said, rags may aberrantly cleave and cause translocation in many of those patients. But then the fact is, normal people, like healthy people like us in B and T cells, we can have rag expression too, right? So then how are this process going to get regulated? Otherwise, everyone will get translocation. Everyone will have all those problems. So there are different layers of regulation already people have described, like epigenetic and many other things. So now we recently added one more layer to that. That's where we found out that there's a microRNA. I'll not get into this detail at all, called MIR29C, which can regulate RAG expression. That was one of the first reports as far as RAG expression is concerned, regulated by a microRNA. But more recently, we found out that not only 29C, even 29A can regulate. Now, this is a very interesting story, but unfortunately, I'll not have enough time to explain that, because our original finding was only 29C. But sometimes, reviewers force you to do experiments. Although when you get the reviewers comment, many a time we become like quite unhappy, because sometimes, a lot of time they can be unreasonable. But this was one of the case where we got benefited, because they wanted to look in a micro 29C knockout animal, what is the level of B and T cells. Unfortunately, India generating those animals, it will take a lifetime, and PhD students for 5 years, they'll say, no, it's a good time for me to leave. So those kind of experiments, sometimes it's very difficult to do. Fortunately, one European collaborator, or the other way, he became collaborator later, had this animal, and he generated this for diabetics studies. So we asked him, can you share this? But again, bad luck. His study was completed, and he has eliminated the colony itself, But in his paper, he had analyzed B and T cells for other purpose. So they found there is a reduction. Okay, there's a reduction when 29C is mutated. So our reviewer was happy. Okay, paper got published. But then we started looking his papers carefully. So we saw that 29A also he generated animal, again for something else, right? So then we looked at the level of this B and T cell, those animals, they showed much better phenotype. So, we started working reverse. So that's the origin of this study. Mm. So this I'll skip. And coming to the second portion, okay, so to summarize whatever I said, I know I'm telling too many things, so hopefully you are not getting confused. So far I said just a little bit about translocations, and I talked about sometimes the sequences playing uh, a causative factor for the fragility. Okay, that's one of the point I said. Then I also talked about there are certain proteins like RAGs, which is normally required for B cell T cell rearrangement, can also cause like misrecognize some of those structures, cutting it. And there's another enzyme called AID and different types of DNA structures and things like that. So that's what I'm right now. So now we'll go to the second part, which is that's where Sheetal initiated a lot of work in the lab. Mm. About the DNA double-strand break repair itself. So now, just to introduce, probably you'll know there are different DNA double-strand break repair pathways. The most common or most, I would say, famous is something called homology-directed repair, and the other one is NHEJ or non-homologous end joining. I'm sure many of you would have heard about this, right? So now, end of the day, either this way or that way, integrity of the genome is maintained. So that means continuity of the genome is maintained. Chromosomes are intact, no translocation, nothing, it's fine. But there is a problem. The problem is if it's NHEJ, which actually take care of more than 70% of breaks in our cell, okay, uh, it's not very accurate. It's not very precise. Okay, HR homologous recombination after repair, you will get back exact same sequence what was there. But NHEJ, there can be little insertions, deletions, some changes can be there, right? Then you may wonder, oh, if 70% of the breaks are actually handled by that, so how are we surviving, right? And when that is a major pathway in human and other mammals. We can discuss that towards the end if you have questions. So now the interesting aspect is good or bad. All these pathways are evolved with, there are so many players, so many proteins, helps in repair of that. I'll not get into all those things because it will become very boring. But NHEJ is what I'm going to cover quite a bit today after this. Therefore, I need to explain that. That's this middle pathway. So I'm sure you would have heard about protein called Q70, Q80. Many of you again would have heard. This is a DNA end-binding protein. Whenever there is a break in the nucleus, this protein, it's like a histones, it's like a present in huge amount in the cells. It will get recruited to the DNA ends. And then once it binds to the broken DNA, this protein recruits the rest of the protein machinery. Like, for example, DNA PK, which is a kinase. Then there is Artemis, which is a nuclease. And this together, Artemis kinase together, it acts as a endonuclease. So these enzymes help in processing of broken DNA because the damage, as we discussed before, it can be because of many factors. So then if there is a synthesis required, there are polymerases called pol mu, pol lambda. These are also called error-prone polymerases. And then finally, the ligation is done by ligase. Right? Ligation, of course, I am sure you will know, which is a recreation of phosphodiester bond, which means the broken DNA is finally joined. So now, during Sheetal time, yes, Sheetal, I hope you remember this finding. So we also happen to contribute quite a bit related to an alternate pathway of NHEJ, which is called microhomology mediated end join. In fact, 2007, 18, 7 and 8, there were, I think she joined as a PhD student in 2007, right? 7 or 8, there were 2 back-to-back Nature paper published, wherein they were talking about generation of translocation using a alternate repair pathway. And then we got into, and then I have one slide on that. Okay, so you can see this is her first paper from the lab. So what we did here is we come up with an assay system which can be used for studying NHEJ. So now what she did here, the see, we mimic the DNA breaks by using specific oligomers. So those ends will be mimicking the endogenous DNA break. Okay. Then incubate with the cell-free extract, which currently she is continuing as well, which you know, cell-free extract means all cellular protein, but RNA DNA is removed. Okay, so you take out the cell total protein, incubate with this DNA substrate, which containing breaks, and then try to look for the joining as shown here. And this can be read in different ways depending on what kind of model system and what kind of facility you have, right? So at the end of the day, NHJ is nothing but end-to-end joining. So if two molecules join, it is a dimer, trimer, three, like that it goes. Okay, Sheetal found out that the NHJ efficiency varies in this she used rat as the system. And she found out that germ cell testis shows the maximum NHJ efficacy, whereas brain, thymus, spleen, lungs efficacy is good, but not, but compared to heart, liver, and kidney, I mean, but in the case of heart, liver, and kidney, the efficacy was much poor. And she went on to show many other results, but I will not be getting into those details. And Satish, other PhD student in the lab, showed that in cancer cells, the BCL2 can interact with Ku protein, and which can downregulate the NHJ. And then if you look at the expression of BCL2, you see here, wherever BCL2 is high and the level of NHJ was downregulated. This is more recent study. There is another protein called RNase H2, which is normally responsible for cleaving RNA DNA hybrid. Now, there is another paper, a science paper showed where RNase H2 play a role in NHJ by Ramsden, Dale Ramsden. Right? So what Dipayan did was he depleted RNase H2 from leukemia cells, and then what you can see is accumulation of the DNA breaks as shown here using the, I think it's a gamma H2 assay, this foci, whatever you are seeing, right? Which means this can be used as a potential target to develop inhibitors. So as I said, when Sheetal was there, she tried to modify this NHG assay system by changing the sequence. So when you introduce direct repeats, so which is also called microhomology, then the DNA repair machinery completely switches. Then the kind of protein used in this alternate pathway is something totally different. Now, if you ask us, it was all discovered from our lab? No. It was partially found by us, but there are many other groups also contributed. And but again, when we tried to publish this work, we had a lot of difficulties. I hope, Sheetal, you remember. We submitted several places. It got rejected one by one. And then finally, it got published here, which is, I would say, a decent journal. But we were trying to, publish somewhere else. But the good news, probably you have seen, this paper is cited more than 200 to 20 times or something like that. So what is the meaning of that? Which means other people found your work more acceptable to them, right? So that was something very good. Whereas our RAG papers, if you look at it, it's like 20 citation, 30 citation, 50. Yeah, I mean, it's like sometimes, it's difficult. Okay, so this is another study where we found that a lot of mitochondrial deletions seen in patients actually go through something, the same MMEJ pathway, whereas NHJ in mitochondria. So I'm sure you know, like in nuclear genome, we have mitochondria, and mitochondria has also a separate genome, which is very small, like 14 kb, whereas other human genome is like huge, right? But what we found out here was the direct repeats are used to repair the breaks in many of those cases, but the major stability of the genome is maintained still by homologous recombination, even in mitochondria. While working on that, we also identified a new function for endonuclease G. Now, what is endonuclease G? I know I am keep bringing up new terms, new enzymes, new protein. Endonuclease G is normally associated with apoptosis, right? The final step of DNA fragmentation, endonuclease G cleaves the 200 base pair distance. So now what we found out was in the mitochondrial genome, this endonuclease G can enter first of all to mitochondria, and it binds to the mitochondrial genome at the G quadruplexus region, and that helps to generation of one of the most common deletions in mitochondria, which is called a 9 base pair deletion. I mean, probably you are not aware, but if you are a mitochondrial related, mitochondria related researcher, you probably would have heard this. Okay, so now I'll skip this. This is still something unpublished and very recent work on NHJ from the lab. So I'll just summarize that. What we found out is, see, if you look at literature of DNA double strand break repair, like what I said, HR, NHJ, MMEJ, these are all well established. There is also something called SSA, single strand annealing, which I did not touch upon much. But now what we are finding is, actually, even the so-called classical NHJ is not sequence independent. But again, if you look at all the papers published, you might read in a different way. It says like irrespective of sequence, NHJ work. That's what generally you read. But here, I mean, I have a lot of data, but unfortunately, again, we have to finish in a little more time. So I'll skip those. But what we found out was, if there are AT-rich regions, and if there's a break at that region, the way NHJ operate is different. Okay, so which is something called coup independent NHJ. The classical NHJ is always dependent on coup. I have explained, whenever there is a break, coup get recruited first. But in this case, if the break is in a AT-rich fragile region, then there is no coup recruitment. Like S4XRC4 directly can go and bind. This is something we are trying to publish. Let's see where it's going to go. Okay, so now we are reaching to the last section, which is what Professor Banerjee was. Anyway, a little bit he has already covered, so my task is almost completed, but still I'll just a little bit explain about the whole story. Now, why would you target NHJ? Right, NHJ is something in a healthy cell, and it's very important. Why would you target NHJ? This can be always a question, and that question I'm still facing from companies. So that's why SCR7 taking forward, we had difficulties as well. So now the logic is, if you look at several of the cancers, DNA repair pathway is deregulated. And many cases, actually, it is upregulated. I will also show you a slide later that, for example, ligase 4 is upregulated in 50% of oral cancer patients. Okay, so I'll come to that. So there is an advantage because of the fact that NHJ is upregulated in many cancers. So the question we asked was very simple. Can we block the NHJ in cancer cell, right? So if you can do that, the result will be something like what I said. When you do the chemotherapy, radiotherapy, as I said before, it all induces DNA damage. So here the idea is use a small molecule inhibitor, a chemical inhibitor, block the NHJ such that NHJ in the cancer cell, can't progress. You will have a lot of, breaks and accumulation of DNA break, which may activate the cell death pathways. So again, it's a long story. So Mrinal Srivastava, again, I think she's Shidal's junior. So these 3-4 students I got same time, and actually they really made the lab. See, when we are a young investigator, I'm sure some of you may be, we are always, at that time I was really worried and scared how my lab is going to be. See, I am coming from PHU, going to IIC, whether I'll be able to guide these people. The students also think they are super smart and all those things, right? So always that tension is there. But I think I was fortunate to get 2, 3, 4, 5, 6 very good students my early career. So they really built the lab. Now, see, current students are also good. Current students are also good. But, always there are a lot of things. Yeah. That's a different area of discussion. But anyway, let's get into this. Right. So what Mrinal, so she's now a faculty in TFR Hyderabad. What Mrinal found out was, by doing lot of experiments, she identified a molecule called ACR7. Okay, it's a chemical inhibitor. Now, so in this, there are multiple collaborators involved, chemistry, then of course, computational biologist, then, animal biologist, many groups who are actively involved. So that's where an institution like this, you have a lot of advantage. Because see, place like IISc, although we claim that we are the best and all those things, there are many limitations. If you have to have a clinical collaboration, no, it's not like that I don't have or others don't have. They also have, even I have, but it's very tough. Actually, I'm honestly telling. Whereas in your case, just you need to walk like what, 20 meters, 50 meters. That's all. So see, current research, especially, current, I mean, about India government, current policy and all. No? So we are looking for the translational research and to have some sort of product from everything what we do. Honestly speaking, that's very difficult, right? Basic research, then you take forward to translational. But an institution like this, where you have accessibility, see, the problem for people like us is, if you noticed, oh, we are good at biochemistry, somewhat molecular biology do well, cell biology also we do well, and maximum now you will see in the next section we'll use a lot of animal models also, but when testing in human it's actually very difficult, right? So whereas a place like here, if you can really remodel, rethink, and I'm sure you are doing already, you can really take the research to next level, like what Western countries people do. See, if you go to countries like Europe and US, almost every building, every hospital, every research institute have a, like, it's all like one floor is research institution, other floor is hospital. Even sometimes you have animal house also. Like wherever I worked, it was a Norris Cancer Hospital, Los Angeles. So for first floor, 4 floors is hospital, cancer hospital. Then fifth, sixth, as much as I remember, seventh, I think it was a research lab, and then half of it was an animal facility too, right? So that's the advantage you have, and then I'm sure you will be utilizing that and taking advantage of that. Okay, so we have come up with this molecule ACR7, which this is what Dr. Banerjee was telling. We published in 2012 in Cell because it was the first NHJ inhibitors to be reported. And then we also showed that it has the potential to, kill, let me tell in layman's language, kill the cancer cells and then help in regressing tumor in animal models and all those things. Now, so whenever you publish big papers, please understand, especially from countries like us, people will be behind you. You have to be very, very careful. Okay, that's another big story. I can tell some other time. But then what we found out is ACR7 can get cyclized into another form, which is called ACR. Parental ACR7 can get cyclized into ACR7 and then to pyrosine. It can get oxidized into pyrosine. But all those forms are active. Like Dr. Banerjee said, I also have a slide. So our molecule is sold by so many companies around the world, only as a biochemical inhibitor, and also it has a use in CRISPR-Cas9 mediated genome editing. I don't want to get into that. CRISPR-Cas9, again, genome editing at a specific site, right? Cas9 induces cut. Then the cell is always dependent on, sorry, either NHJ or HR, homologous recombination, for the final editing. Now, if the HR operates, then then you will get a precise genome editing. Otherwise, it will be imprecise genome editing. What people have done, two groups, one from Germany and other from US, so MIT actually, they used ACR7 in their CRISPR-Cas9 system to show that the precise genome editing can be improved significantly and so on. Okay. So that was all good stories, and it's patented and all those things. But the bad part was, it has limitations. One is the IC50. So that's what I was talking to companies. They were not very excited about our molecule. One is it's a DNA repair inhibitor. Second thing is IC50 was quite high. So that's where we started derivatizing this molecule. Again, I'll not get into the details. Now, whatever I'm going to show mostly is unpublished. So we have derivatized more than 180 molecules newly synthesized. And we do this NHJ screen, which is established by Sheetal and others later in the lab. And then you can, once you screen and identify which molecule work good and things like that. And now we have two molecules called SCR116 and 132. This is also patented, both India, US, and all those things. And if you look at the ligase for knockout cells, the efficacy is much reduced. The first part is published. Now we are characterizing this molecule further. And now if you treat with increasing concentration of this molecule, you can see the DNA breaks there, and then several cancers it work. The efficacy is anything between 50 to 100-fold higher than ACR7. I think I'll skip this. Okay, so this is something interesting. So that's, we have done animal studies, and now ICMR is supporting this project in a very big way, and we are taking forward. Now the studies are going on with the new molecule in GLP certified lab, but still is early stage. Now, the one aspect which we want to focus with this molecule is this. So this is the thing which I was telling at the beginning, oral cancers. These are RNA-seq data from abroad and some from India. There are about 37, 40 patients data RNA-seq oral cancer from India. So what you see here is very interesting. About 50% of these patients have ligase 4 expression quite high. ACR7 actually, maybe I missed to point out that ACR7 actually target the ligase 4. Hmm? So that's where we are proposing to use. See, this is from cell line. Again, you can see 50% of the cell lines what we have, and this is the Western blot. See, look at the cytotoxicity, and this is the xenograft model, CDX model, where you see the good effect. But this effect is not as good as what I showed for the allograft and things like that. Hmm. So the I think I'll skip this. These are all the new studies going on with respect to the toxicity. See, if you want to take any molecule forward, right? As per CDSCO guideline, DCGI, Drug Control of India guideline, you need to have a lot of toxicity studies. After efficacy, you have to go for toxicity, which can be acute, chronic, then genotoxicity and so on. So that is what currently we are doing as far as that part is concerned. It's now 4:04. Can I take 2-3 minutes more, or is that okay? Okay, so if you allow me, this last section I'll tell, because this is the most promising result, and hopefully I hope something will happen there. Okay, but this is totally unrelated to DNA repair. Hmm? So as I said before, I think this aspects you probably already know. There's a problem with those slides. So BCL2 is upregulated in majority of the cancers. And if you, I think you guys will be, many of you will be clinicians. You will know that there is only one FDA approved BCL2 inhibitor so far in the world, which is called ABT199 or venetoclax. And there were many efforts by several people to develop BCL2 inhibitors. Okay, so now if you remember, I was working on those BCL2 translocation during my PhD, which is follicular lymphoma. So that's like long time back. But when I joined as an independent PI, I was trying to contact chemists, asking, can we collaborate? Can we think about developing a molecule which will be targeting BCL2? So that at that time, my thinking was only with respect to follicular lymphoma, where BCL2 get overexpressed because of that translocations, right? But you know that actually majority of the cancers actually BCL2 is overexpressed. And then so our research started somewhere around 2008-9 with respect to this. And then many things happened because of the many reasons, students and news people. Sometimes it got delayed and things like that. But during the course of time, okay, so by the way, so we have our BCL2 inhibitor with the help of, I think I did something wrong. Okay, um, which is called Dicerib. Again, we collaborated with medicinal chemists, and many were synthesized and screened against cancer cell lines. And what you can see is this molecule Dicerib, or 5M, which showed BCL2 dependent cytotoxicity. That was our first hint. Then what we did is we tried to knock down, we tried to overexpress BCL2, then try to look at the level of expression. We also used BCL2 knock out cells, and once incubated with the BCL2 inhibitor, you can see the knockout cell become insensitive. Then what you do? You do standard docking studies in silico. There are other anti-apoptotic genes like what is mentioned here, BCLXL, and also BCL2A1 and many of those, right? Then we purified the proteins, did the biophysical experiment to show that indeed the chemical interact with the BCL2. And now if you look at the BCL2, it has different domains, BH1, BH2, BH3, and transmembrane domain and all. We try to mutate one by one. So now what you see here is it predominantly interact with the BH1 domain. Okay, so this is a story up to say 2013-14. So somewhere around 2016, first BCL2 inhibitor got clinical approval, which is what I said, venetoclax. So now if you look at the BCL2, the venetoclax, that predominantly bind to BH3 domain of the inhibitor. So there was a difference. But again, we were doing a study But then we were not expecting something, so this will become one day something promising or something like that. Some extent, again, reviews helped. See, we got the cell paper, so we had decent data on this inhibitor. We tried to send to Nature Medicine and big journals, but unfortunately, they all kept 1 month, 2 months after that, they rejected. Okay, so finally, we decided to split that and publish in, I'll show you that, 3 different journals, which is like somewhat low profile journals. But one good thing came from all those activity or process was one of the reviewer insisted us, you compare the efficacy of inhibitor with the venetoclax. Okay, but then the point was we had already compared this. I have a collaborator from UK. He's a hematologist. He had a good friend of mine. He used to tell, "Oh, we have a BCL2 inhibitor. It's just going to get approved and all those kind of things." And so he gave a aliquot, but we compared, we tested that and compared, and then we found, oh, our inhibitor is better than that, which honestly we did not believe for 1-2 years. But when the reviews insisted us to test this, we had to, and then we had to buy a new aliquot. That's where this whole study become kind of, you can see here, when we compared, we found that our inhibitor worked better. And this is animal model, cell line. And after that, this was repeated in several animal models. Okay, and it was published that time. And now this inhibitor we have tested in about 13 different animal models, xenografts. So it worked decent. And then we have also looked at, in collaboration with Kiran Mazumdar Show Center Hospital in Bangalore, around 123, 126 patients we have tested. Somewhere around 65% of the cases, this inhibitor showed effect, which is comparable to venetoclax, which we directly compare with venetoclax. And then I think I'm going to skip all those things. Okay, so this is now the preclinical toxicity studies, like what I was telling, it's all completed in rodents and non-rodent rabbits. We are in the process of filing the IND application. So if that goes through, then hopefully we'll be able to go for phase 1 clinical trial. And so now our molecule is in, see, formulation studies are going on. It's in the tablet form, as you see. But of course, it requires many more processing and things like that. As far as BCL2 inhibitor is concerned, we are now here, very close to phase 1. Let's see how our IND application is going to go through. And actually, thank you very much. I thought you all will run away after 20 minutes, 30 minutes. But since all of you sat, I got really, inspired more and talked. I think it's around 45, 50 minutes talk. I'll be very happy to take any questions you have. But before that, let me thank my group. Many of their names I have already taken. And several of the collaborators, and of course, generous funding from DBT, DST, DAE, ICMR, and several other sources. Really, really thankful for your patience and then being here and spending time. I'll be happy to take any questions you have. नहीं ले रहे. Okay. Shidil is telling इसमें questions नहीं लेते, so we can talk in private later. Thank you.
Thank you, sir, for such a wonderful, inspiring, and knowledgeable talk. I'm sure we all learned a lot about how DNA is broken, repaired, and how can this process be utilized for cancer therapeutics. May I now request Professor Sanjay Jain, Professor Dibyajyoti Banerjee, senior faculty members to kindly come and felicitate Professor Raghavan. Thank you, dignitaries. We are honored and thankful to have late Professor R.N. Chakravarti's family's message for this event. Good afternoon, everyone. For many of you who don't know me, my name is Ranjan Chakravarti, the eldest son of Dr. R.N. Chakravarti. On behalf of our family, thank you all for being here today. It is truly moving to have so many leading physicians, scientists, and research scholars who knew Dr. Chakravarti not just as a colleague, a doctor, and a dedicated scientist, but also as a mentor, sincere friend, and a devoted family member. He loved medicine, but more than that, he loved people. He always said that being a physician was a privilege, not just a profession. We feel truly humbled when his work is recognized for the quality, rigor, and dedication in this August Institute devoted to the advancement of science for improvement of care for the common man. While we miss him deeply, knowing that this memorial lecture will honor his memory and inspire future generations of physicians and scientists brings us tremendous solace. We are honored by the presence of Professor Satish C. Raghavan today. Thank you, sir, for delivering this year's lecture. Please enjoy this time of remembrance and learning. Welcome, everyone. Thank you, sir. Uh, I now request Professor Alka Bhatia ma'am to kindly extend a vote of thanks.
So, Dr. Satish C. Raghavan, Department of Biochemistry and Chair, Central Animal Facility, Indian Institute of Science, Bangalore. Dean Research and Chief Guest for today, Professor Sanjay Jain, and Professor Divyajyoti Banerjee, Head Department of Experimental Medicine and Biotechnology, PGI Chandigarh, Professors Emeritus, faculty colleagues, staff members, and dear students. At the close of today's intellectual feast, it is my duty to extend or propose a vote of thanks to all those who have contributed in one way or other towards this event today and made it a huge success. I congratulate you all. I, on behalf of the Institute and Department of Experimental Medicine and Biotechnology, extend a hearty vote of thanks to our esteemed speaker, Professor Raghavan, who shared with us his wonderful knowledge and remarkable journey in the field of DNA double stranded break repair and its implications in cancer therapy. We understand, sir, that you have taken time out of your very busy schedule to be with us here today. And thank you for your insightful talk. I must mention my deep sense of appreciation towards Professor R.N. Chakravarti's family, who have sent their remembrances and kind wishes on the occasion. I also wish to express my gratitude towards PGI authorities, Department of Telemedicine, Photography, Horticulture, Sanitation, members of the organizing committee, and all those who have worked behind the scene tirelessly to ensure smooth running of this event. This vote of thanks would not be complete without acknowledging the august audience present here today for their time and attention. At last, I conclude this session with sincere thanks and anticipation of such kind of interactions in future. So as they say, we part until we meet again. Thank you.
Thank you, ma'am, for your kind words. Sir, we need your kind permission to conclude this event. Thank you, sir. We conclude this oration and request all of you to please join us for high tea in the foyer. Thank you all.